Okay, good morning class. Okay, we are going to talk about the cell physiology, which is our second topic. So when we talk about the cell physiology, we are going to this is the study of the functioning of the cell structures. So first we're going to look at what do you mean by the cell. So as you can see that it's the smallest structure and functional unit of living organisms, which is often referred to as the basic building blocks of the life. Cells can vary in size, shape, and function, but they are share a common characteristics. Then, cells are enclosed by the cell membrane, which separates the cell's interior from its external environment. So we are going to look about the two primary types of cells, whereby we have the prokaryotic cells and the eukaryotic cells. Starting by the prokaryotic cells, so these cells lack a nucleus and membrane-bound organelles. They are typically found in simple organisms like bacteria and archaea organisms. Secondly, we have the eukaryotic cells. So talking about the eukaryotic cells, they have a defined nucleus and membrane-bound organelles. Then they are found in more complex organisms, which includes plants, animals, fungi, and the protists. Cells are the basic units of life and work together to form tissues, organs, and ultimately entire organisms. We can say they can carry out essential functions that sustain life, such as growth, reproduction, responding to stimuli, and maintaining metabolic processes. So next I want to talk about what is what is physiology. We said that physiology is a branch of biology that focuses on the study of how living organisms function. Physiology is a branch of biology that mainly focuses on the study of how living organisms functions. So it explores the mechanisms and processes that enable organisms to carry out vital functions and maintain their internal balances. That is what to call homeostasis. Then physiology seeks to understand how biological systems work, including the processes like digestion, respiration, circulation, muscle contraction, nervous system function, and more. So this knowledge is crucial for gaining insights into health and disease, as it provides a foundation for understanding how the body functions under normal and abnormal conditions. Physiological research is essential in fields like we have medicines, sports science, and biology to improve our understanding of living organisms and their capabilities. Cell physiology is a branch of biology that focuses on the study of how individual cells function and carry out various processes. So it encompasses the investigation of cellular activities such as metabolism, cell division, signaling, transport of molecules, and responses to external stimuli. Next one, cell physiology is a fundamental field of biology that delves into the intricate working of individual cells, the building blocks of life. So it explores how cells function and maintain the conditions necessary for life. Then now see the properties of the cell. We have now talking about the membrane structure and the properties. When we talk about membrane structure, you say that it's a surface structure which encloses the cell and organelles. The membranes regulate the flow of material in and out of the cell. For examples of membrane, we have the cell membrane, we have the tonoplast, we have the nuclear membrane, and the mitochondria. So let's start by the cell membrane. We're going to go to see the structure of the cell membrane. Say it has three layers, which is the two protein layers and a phospholipid layer sandwiched in between the two. That is the in between the two cell membrane. Then now let's see the properties of the cell membrane. One, we said that it is semi-permeability. So it it has small pores allowing for the passage of molecules of small size into and out of the cell. Cell wall, however, allows all materials to pass through it, hence it is referred to as being permeable. So when we talk about semi-permeability, this is whereby the cell, it has the ability to allow selective movement of materials in and out of the cell. Then we say have the next one is say sensitivity to changes in temperature and pH. Sensitive to changes in temperature and pH. Extreme temperature and pH affects the cell membrane since it has some protein layers. Such changes alter the structure of the membrane, affecting its normal functioning. Another property of the membrane is that possession of the electric charges. So it has both the negative and positive charges, helping the cell to detect the changes in the environment. The charges affect the manner in which substances move in and out of the cell. Then let's see the physiological processes. The ability of the cell to control the movement of substances in and out of the cell is achieved through physiological processes such as diffusion, osmosis, and transport. Then I started by first physiological process is diffusion. So diffusion, you can say this is the process by which particles move from a region of high concentration to a region of low concentration. So diffusion is a process by which particles move from a region of high concentration to a region of low concentration. Then the difference in concentration of particles between the region concentration and the higher concentration flow concentration is known as diffusion gradient. So when you talk about diffusion gradient, this is whereby is the difference in concentration of particles between the region of high concentration and the region of low concentration. Then let's now see about the roles of diffusion in living organisms. We have absorption of materials. Mineral salts in the soil enter the root by diffusion, since their concentration in the soil is greater than in the root air cells. So the digestive food, that is, you have the glucose and amino acid diffuse across the cell wall of the root into the blood for the transport to take to the rest of the body. Then, so talking about the role of diffusion, we have absorption of materials. When we talk about the absorption of the material, this is whereby plants obtain mineral salts from the roots by a process known as diffusion. So this is the first primary function of diffusion, that one which normally is functioning in the roots or in plants. Then another role of diffusion is gaseous exchange in plants and animals. So in plants and animals, respiratory gases, that is oxygen and carbon dioxide, are exchanged through simple diffusion depending on their concentration gradient. So talking about the gases exchange in plants and animals, this is whereby respiratory gases, that is oxygen and carbon dioxide, they are exchanged through simple diffusion depending on their concentration gradient. So let's see factors that affects diffusion. Factors that affects diffusion. 1. Diffusion gradient. So diffusion gradient between 2 points increases the rate of diffusion. Surface area to volume ratio. So another factors affect diffusion, you have surface area to volume ratio, whereby we say that the higher the ratio, the greater the rate of diffusion, and the lower the ratio, the lower the rate. This means that small organisms expose a large surface area to surrounding compared to large organisms. Small organisms therefore depend on diffusion as a means of transport of foods, respiratory gases, and waste products. So when talking about the diffusion, the surface area to volume ratio, this is whereby we can talk now about you compare the different types of organisms, whereby we have, say for example, we have the higher organisms, there's large organisms and the small organisms. So the higher the ratio, the greater the rate of diffusion, and the lower the ratio, the lower the rates. So this means that small organisms expose high surface area to the surrounding. For example, we can talk about the elephant that exposes small organisms surface area to the surrounding, and then compared to large organism. And then you have small organisms therefore depend on diffusion as a means of transport of foods. And then we have respiratory gases and waste products. Then you have the thickness of membranes and the tissues. Thickness of the membranes and the tissues. So the thicker the membrane, the lower the rate of diffusion, because the distance covered by the diffusing molecule is greater. The thinner the membrane, the faster the rate of diffusion. So talking about the thickness of the membranes and the tissues, whereby the thicker membranes, they have the low rate of diffusion because the distance covered the diffusion molecule is greater, while the thinner the membrane, the faster the rate of diffusion. Then talk about the size of the molecule. Small and large molecules diffuse faster than large and heavy molecules. Small and large molecules diffuse faster than large and heavy molecules. So let's see the next one we call the temperature. An increase in temperature increases the energy content in molecules, thus causing the more particles to move faster. Then we have the osmosis. Now we have talked about the physiological process, but we are talking about the diffusion, we talked about the osmosis, and then talk about the active transport. And under the diffusion, we are talking about this is the process by which particles moving from a region of low concentration to a region of higher concentration, and we have listed some of the factors that affects osmosis diffusion. One, we say that we have the diffusion gradient. Second, we have the size of the molecules, and then we talked about size of the molecules and the size of the membrane of the tissues, and then we have the temperature. Then next type of physiological process we have osmosis. So talking about osmosis is a process where solvent molecules, e.g. water, move from a lowly concentrated solution, that is a dilute solution, to a high concentrated solution across a semi-permeable membrane. Then I repeat, talking about osmosis, it is one of the physiological process whereby it involves where solvent molecules, most likely the water, move from a lowly concentrated solution, a solution that has low concentration, to a highly concentrated solution, therefore across a semi-permeable membrane. So here we define some of the terms here. We have hypertonic solution. Talking about hypertonic solution, which means that it is a highly concentrated solution. Then we are talking about hypotonic solution. This involves a solution of low concentration. Then last one we talked about the isotonic solution. An isotonic solution is a solution of the same solution. Then we can say osmosis as a special type of diffusion because it involves the movement of solvent molecules from the region of high concentration to the region of low concentration across a semi-permeable membrane. This is why we call it a special type of diffusion. You can use an example, for example, when you use a whisking tubing, whereby you can just say if you want to test for the starch in that kind of a whisking tubing, whereby we put the starch content inside the whisking tubing, and then you tie the both ends. Then therefore you will put that content in the whisking tubing in a highly concentrated solution, whereby we'll have to find that the content in the whisking tubing will have to turn its color, therefore meaning that the highly concentrated solution has entered the whisking tubing, whereby it will change its color to be blue. Then next what I'll talk about the osmotic pressure. So this is a pressure which needs to be aligned to a solution to prevent the inward flow of water across impermeable membrane. So this is the pressure needed to nullify osmosis. So osmotic pressure is measured using the osmotic potential. So osmotic pressure can be measured using an osmotic potential. So let's now define what is an osmotic potential. So an osmotic potential is the measure of the pressure of a solution that will develop to withdraw water molecule from pure water when separated by a semi-permeable membrane. Then let's see what we call water relations in animals. Relationship between water and the animals. So cell membrane of the animal cell is impermeable, just like the dialysis or pisking tubing. Cytoplasm contains dissolved sugars and salt in solution form. Then here we are talking about now when an animal cell, e.g. a red blood cell, is placed in distilled water. When we talk about distilled water, we could talk about a hypotonic solution. A hypotonic solution is a solution which is lowly concentrated. So then if you take an animal cell and you place it in that kind of a solution, which is a hypotonic solution, we say that water flows in by osmosis. Now the cell will gain water by a process known as osmosis, whereby the cell will swell up and eventually burst because the cell membrane is weak. The bursting of the red blood cell when placed in a protein solution is called as hemolysis. So when we talk about water relation in animals, here we are going to compare the water, how is water related to animal cells. For example, we're talking about if an animal cell, e.g. a red blood cell, is placed in distilled water, but we say distilled water is a hypotonic solution. We say that the water will have to flow or will have to move in to that kind of the cell by osmosis. Then the cell will swell up and eventually burst because the cell membrane is weak. Then the bursting of the red blood cell when placed in a hypotonic solution is called hemolysis. So when a red blood cell bursts when placed in a hypotonic solution, that is termed as to be hemolysis. Then if a similar red blood cell is placed in a hypertonic solution, now a highly concentrated solution, we see that water will be thrown out of the cell by osmosis. The cell will have to shrink by a process called as crenation. Then body fluids around the cell must therefore have same concentration as that which is found inside the cell. So here we have two terms that we are going to differentiate very keenly. Starting by the first one, we have hemolysis. We define hemolysis as we say that this whereby when an animal cell is placed in a hypotonic solution. When we take an animal cell and place it in an hypotonic solution, we'll find that the cell will eventually have to burst. This is because the cell is weak. So now, when the membrane of the cells are weak, it means the cell will gain water by osmosis, and then the content of the red blood cell will have to enlarge, and will have to increase in size, and finally it will have to burst in a process known as hemolysis. That's what we talk about hemolysis, whereby an animal cell is placed in a hypotonic solution whereby it will have to gain water by osmosis, and then it will have to burst. And then at the time we have what we call the crenation. When we talk about crenation now, is whereby we have an animal cell now placed in a highly concentrated solution, which is most likely to be a hypertonic. Now we say that water will have to be thrown out of the cell by osmosis. Then the cell will have to shrink by a process called crenation. So that is the difference between crenation and hemolysis. And the only difference mainly comes whereby when you have the same types of animal cells and you place them in different type of solution. One can be hypotonic and another one can be hypertonic. Then let's now see water relation in plants. Water relation in plants now. Here we are going to talk now how plants relate to different water solution, how to different kind of water solution. So when a plant cell is placed in a hypertonic solution, it will have to gain water by osmosis and distance outwards. As the cell gains more water, its vacuole enlarges and exerts an outward pressure called a turgor pressure. As more water is thrown in, the cell becomes firm and rigid and is said to be turgid. Now, the cell wall in plant cell is rigid and prevents the cell from bursting, unlike the case in animal cells. So the cell wall develops a resistant pressure that pushes towards the inside. Now, this pressure is equal and opposite the turgor pressure and is called the wall pressure. Now, when talking now about the relation in animal cells, which is not in animal cells, when you take a plant cell, you place it in hypotonic solution. Now, a hypotonic solution, you say that is a solution that is slowly concentrated. Thereafter, we will find that it will have to gain water by osmosis, and finally, it will distance outwards. So, this one is the opposite with the diffusion. When talking about now the animal cells placed in this kind of a solution, whereby we find that when you take an animal cell, you place in hypertonic solution, it will have to gain water by osmosis. After gaining water by osmosis, therefore it will have to pass out. Now, when it comes to in the case of the plants, when the same type of plant cell now it's exposed in the same type of a solution, for example the hypotonic solution, we'll find that this type of a plant cell will have to gain water by osmosis and will have distance outwards. So this process of distance outwards, it will create what a pressure that is called a wall pressure. Now, when we talk about the wall pressure, this is the pressure that normally pushed or outwards after a plant cell has gained or has absorbed water, and then the cell of a plant cell does not pass. This is because the cell membrane of a plant is rigid. Then, talking about when a plant cell placed now in a hypertonic solution, whereby a hypertonic is a highly concentrated solution, will have the water molecules that will have to move out of the cell. Now, the water molecule have to move out of the cell into the solution by osmosis. The cell shrinks and becomes flaccid. So now here we have now the difference here. This is where the difference comes from. One, we have when we place an animal cells in a hypertonic solution, we will find that this animal cell will have to become rigid and will have to absorb pressure and develops a wall pressure that will have to prevent the cell from bursting. But now when you take that plant cell, you place it in hypertonic solution, this case which is a highly concentrated solution, the water molecules will have to move out of the cell into the solution by osmosis. The cells will have to shrink and becomes flaccid. Okay, you have to remember, you can call back whereby we are talking about the water relation in animals, whereby we talked now. If you take an animal cell, you place it in a hypertonic solution, the condition that the cell of the animal cell will become, it will have to be what we call crenation. But now for the case of a plant cells, it will not become crenation, but it will have to shrink and it will have to become flaccid. If the cell continues to lose water, plasma membrane pulls away from the cell wall towards the center. Now the process through which plant cells lose water, shrink and become flaccid is what we call plasmolysis. Plasmolysis can be reversed by placing a flaccid cell in distilled water, and this process is called wilting. Now, When plants lose water through evaporation and transpiration, the cell loses digidity, shrink, and the plant drops. So this is called, if water supply from the soil is inadequate, plants do not recover, hence permanent wilting. So that is what we call permanent wilting. Now talking about wilting, when plants lose water through evaporation and transpiration, the cells will have to lose digidity, they will have to shrink, and the plant will have to droops. This is what we call now wilting. If water supply from the soil is inadequate, plants do not recover, hence it undergoes what we call permanent wilting. Now let's talk about what we call now the roles of osmosis in organisms. Roles of osmosis in organisms. Roles of osmosis in organisms. 1. Osmosis, it helps in the absorption of water from the soil. Osmosis helps in the absorption of water from the soil by osmosis. So the roots of the hairs of the cell or plants absorb water from the soil by osmosis. So if you have to find that we have plants, someone may ask, how do the plants obtain water or nutrients from the soil, and hence they don't have even what to feed from? But this one is normally carried by the help of what we call osmosis, whereby we say osmosis, it involves whereby plants have to acquire mineral salts from and water from the soil by a process known as osmosis. Now the second role of osmosis, we have to talk about support. So cells of herbaceous plants which are less woody absorb water, become vegetarians, the support. Now when we talk about the support, we're talking about the plants which cannot withstand or stand on their own. So this means that they will have to obtain support from elsewhere. For example, of those kind of the plants, we have the herbaceous plants, which we say that they are less woody, whereby they absorb water, become touched, and hence they support. So through osmosis, the herbaceous plants obtain support to withstand their climatical conditions. Then the next one, we have the opening and closing of the stomata. Opening and closing of the stomata is another role of osmosis. So this one, you can say that during the day, the gut cells synthesize glucose that throw in water, become digit, hence open the stomata. During the night, they lose digidity, hence there is no photosynthesis. As a result, they shrink, thus closing the stomata. Now, when we talk about the opening and closing of the stomata, this is kind of the practice normally take out place during the day and during the night hours. So we said that during the day we have what we call the gut cell that synthesize glucose that throw in water, they become turgid, hence open the stomata. So during the night now, we can assume that there is no what we call photosynthesis. So the plants, they lose the rigidity since there is no photosynthesis. As a result, they shrink, thus closing the stomata. This is another exercise whereby plants, they normally open and close their stomata during different weather and climatical conditions. Say for example, when the weather is not favorable to them. Say for example, during the night hour they have to close their leaves, and during the day hours they have to open their leaves. Then you have what we call feeding in insectivorous plants. This is another role of the osmosis. So osmosis helps in feeding of insectivorous plants. So these plants are able to change their target pressure on the leaves, which close, trapping insects which are digested to provide the plant with what we call nitrogen. So when we talk about now the role of osmosis, which is the fifth one, you can see that it helps most likely in the feeding of the insectivorous plants. Now here is whereby we have some of the insects that normally comes to the flower parts of the leaves. They attach themselves on there, and then they continue sucking the sap from the leaves and the flowers. So in this process, there are some of these plants that obtain their nutrients or nitrogen from this kind of of insects. So once the insects lands on a flower, this type of the plant will have to close their flowers. So meaning that this type of an insect will have to act as a food for that kind of a plant. Then another role of what we call now osmosis is what we call osmoregulation. Osmoregulation. So in the kidney tubule, water is reabsorbed back to the body by osmosis. So this osmoregulation normally takes place in the kidney tubule. This is why it involves water being reabsorbed back to the body by a process called as osmosis. Now let's see what we call now factors that affects osmosis. Factors that affect osmosis. 1. Concentration of solution and the concentration gradient. Concentration of solution and concentration gradient. So when we talk about the concentration of solution, we talked about hypertonic solution, hypotonic solution, and isotonic solution. And under the hypotonic, we say that it's hypotonic is a solution that is lowly concentrated. And now talking about the hypertonic solution is a solution that has a lot of concentration. Then talking about isotonic solution, now under isotonic solution is a solution of equal concentration. Therefore meaning that this type of the solutions, when they vary in difference, they have to influence they have to affect osmosis. Another one, optimum temperature, as long as it does not destroy the same permeability of the membrane. So optimum temperature, it normally also affects osmosis in some ways or the other. Now let's now see at the third physiological process, which is now called the active transport. So this is the process that moves substance across cell membrane against a concentration gradient. Talking about active transport, it can be mentioned that it is a process that moves substances across the cell membranes, whereby is against a concentration gradient. Now this process requires energy to move these substances across the cell membranes and involves substances such as amino acid, sugar, and many ions are being taken in by living organisms through the active transport. Then now let's see what we call the roles of active transport. Roles of active transport, starting by 1, the absorption of sugars and useful substance by the kidney. So the active transport plays a very vital role in the human body or in the functioning of the human body. 1, we say that is what we call, it helps in the reabsorption of sugars and useful substances by the kidney. Another one, we have absorption of some mineral salts by plant roots. So active transport, it helps in the absorption of mineral salts by the plant roots. This is whereby these plants will have to obtain the mineral salts by the roots and then through a concentration gradient, which is called now the active transport. Another one, we have absorption of digested food from the alimentary canal into the blood stream. So this is another very vital role of the active transport, whereby it helps in the absorption of digested food from the alimentary canal into the blood stream. The fourth one, it helps in the accumulation of substances in the body to offset osmotic imbalance in the arid and saline environment. Then the fifth helps in the excretion of waste products from body cells. Now let's see what we call now factors affecting active transport. One, we have what we call the oxygen concentration. Oxygen concentration. We then have the second what we call the change in pH. When pH varies or pH changes, the rate of active transport will have to be influenced. And then we have glucose concentration. Then we have another one what we call enzyme inhibitors. So these are the factors that normally affects the active transport.